Hi, everybody. This is Joe Barrascano, Dr. B, presenting for IGeneX. And tonight we're going to be talking about some news in the testing front. Um, First of all, one of the things is that the Lyme IgM ImmunoBlot has received FDA clearance. The IgG Lyme ImmunoBlot has also received FDA clearance. Barnett ImmunoBlots, which are all-time favorites, both the IgM and IgG have now received New York State approval and are available nationwide. And just to remind you, the Babesia ImmunoBlots are also available nationwide. So that's old new news. I want to start by saying that, I've always used IGeneX for my testing ever since I opened in the 1990s. And, the results always seemed to be more reliable, and I trusted them more than from any other lab. And, even back in the day, there were state labs, there were commercial labs of other kinds, there were federal labs even, but IGeneX always seemed to be better. And going through this, I don't work for IGeneX, I just do their public speaking. So I don't really have access to the inside information until now that I've seen what they have gone through to validate their testing. It really is impressive. Also kind of reassuring to me to see really the science behind it and the hard data that shows it really is as good as we have hoped. Now, what does the FDA clearance mean? I'm going to start with that, and we'll go into it a little bit more later on. It just means that these immunoblots can now be performed by any commercial lab. All they have to do is change from whom they buy their test kits. So that sounds simple enough. The kits are manufactured by a company called ID Technology. Now, what is that? Well, just to end confusion, IGeneX is a testing lab, and they're not a manufacturer. So they formed this company. It's the same people that run IGeneX are part of ID, and their test kits are marketed as the iDart. So when you hear an iDart by ID, it's basically the same technology, the same tests as the IGeneX Lyme ImmunoBlots. The IgG kit was cleared by FDA in September of 24. The IgM Lyme kit was just cleared last month, June of 25. They'll be available to commercial labs once they set up the paperwork and reimbursement rates. They say later this year, late in 25. But in the meantime, the same immunoblots are available through IGeneX directly as you've always had them. Now, the clearing process was really interesting when I looked into it. It took them 2 years. They had to get help from a lot of different people, and I'm highlighting both the CDC and the Bay Area Lyme Foundation because CDC, they have a bad reputation in the Lyme world, but the truth is they were really very helpful to the lab in terms of getting the thing set up to get through the FDA, because the CDC and FDA are two separate organizations, or whatever you want to call them. And the CDC helped IGeneX get FDA cleared. So that was really a nice thing. And the Bay Area Lyme Foundation, they supplied samples from their tissue bank, which is in the serum bank, which is really, really great. IGeneX also got help from New York Biologics, Caminiti Life Sciences, the FDA itself, Ward Medical Laboratory. All these other people provide samples or assistance. And IGeneX itself used their own samples for validation. And the result is that this company, ID, which is basically IGeneX, is one of the only a few companies to achieve FDA clearance for Lyme disease test. And it's the only one that's cleared that tests multiple species of Lyme and using recombinant antigens. We're going to go through all this in a minute. Now, one of the reasons why ImmunoBlot is so important and so useful, and many of you have seen this slide before, but I want to emphasize it. Lyme bacteria, Lyme spirochetes, are not a single spirochete. It's a family of spirochetes. The original one, which came from a tick that Willy Bergdorf discovered in eastern Long Island, New York, back in 1980, I believe it was 81, that's been used as the type strain for testing by the government, by universities, by commercial labs. They call it Borrelia burgdorferi sensu stricto, BBSS, and that's strain B31. So lab strain came from a tick, not a human being. Now, all those tests by commercial labs, big box labs, hospital labs, are based on this one thing. It's one strain of one species. The IGeneX ImmunoBlots, however, are not restricted to that one. As you see in the list, there are many, many different Lyme Borrelia spirochetes causing Lyme disease symptoms and illness that are not picked up by these government-backed tests, but they are picked up by the IGeneX testing. Okay, so they call this larger group Borrelia burgdorferi sensu lato, or BBSL. So just keep this in mind, because if you don't test all these things, and I'll show you exactly why later, you're going to be missing your patients. So where did this lab strain come from, this B31? Well, Willie, as I said, Willie Bergdorfer was collecting ticks from Shelter Island, which is a small island 100 miles east of New York City by the ocean. This sample came from a tick, not from a human patient. The basic truth is that this one strain really does not represent the true diversity of pathogenic Borrelia we're seeing in our patients. Borrelia are bacteria that are able to modify their genetic and external appearance based on their environment. So they have to be able to transition from a tick to a reptile, to a mammal, birds, so forth, as well as different climates. And as a result, the different species and different strains are found in different parts of the country. So to take a spirochete from an isolated island on the east coast of America doesn't necessarily represent the full spectrum of Lyme disease that we're seeing in our patients. Now, that's a theory, but it's been proven. I'll show you the data. So the other problem is that they've taken this strain B31 from 1980, 81, whenever it was, and they've kept it alive in culture. Now, it's known for a long time that whenever you keep Lyme germs, Lyme spirochetes in culture for a long period of time, they change. They lose genetic material. They become less pathogenic, and they're very much less like what's really found in nature and in our patients. So now, if you want to kind of characterize the different kind of tests that are available to us, I like to think of them as standard serologies, the old-fashioned kind that we've been dealing with for a while, and then contrast that with the modern ones, the recombinant assays that IGeneX developed. So in a standard serology, they're made of antigens from the germ, and these antigens are part of the test kit that allows them to look for the germ. These antigens are derived from whole organisms, these lab strains that are broken apart. They're lysed with ultrasound. When you lyse an organism, especially a complex organism like Borrelia burgdorferi, many, many antigens are released, some of which are specific to Borrelia burgdorferi, but a lot of them are non-specific. They're found in other bacteria, they're found in some viruses even, and even autoantibodies in people with autoimmune diseases. So when you do a standard serology that's kind of dirty with all these other antigens that are really not specific for Lyme, the way you get rid of so many false positives is you turn down the test sensitivity, okay? So this results in a more specific test, but you get the trade-off is many false negatives, because with these type of old-fashioned tests, there's always a trade-off between sensitivity and specificity. On the other hand, the recombinant assays that IGeneX uses, they're synthetic antigens. They're created in the lab. Now, it's cool how they did this. It's kind of a reverse engineering. So what they do is, let's say they want to get antigen, the 31, the band 31. Okay, what they do is they go to Borrelia, they take the genetic sequence of the Borrelia that makes this protein, they put it in a virus, the virus infects E. coli, which then produces a very pure antigen. So they take this antigen that's now very pure, specific to Borrelia because it came from Borrelia's own genome, will not cross-react with anything else. And then they purify it multiple steps, whatever, and then they make the tests out of that. So this means it's very specific. It virtually eliminates the false positives, virtually eliminates false negatives, and you don't have this trade-off between sensitivity and specificity. The other cool thing is that they can get these genetic sequences from a lot of different Borrelia, not just Borrelia burgdorferi strain B31, but strain 297. They can do other different species. So what they end up doing is coming up with a test, the ImmunoBlot, that can look for a whole wide variety of Borrelia organisms, not just Borrelia burgdorferi sensu stricto, the BB31, but sensu lato, which is that whole list I showed you earlier. So let's talk about the specific test. The old text standard test, which we don't recommend anymore, they don't use recombinant technology. They're using a single lab strain. For example, the IFA, they take the organisms that were in culture, they put them on a slide, dry it out, then they add patient serum. If antibodies in the serum are present, it'll attach to that spirochete on the slide. Then they put a binding human antibodies with a fluorescent dye that binds to it, and they'll look at it under the microscope. It's not very exacting, subject to a lot of technical issues, operator error, it's time consuming, it's really not that great. A lot of false negatives, some false positives too. And again, it's just one species, that thing that they cultured. What came from IFA is that they've advanced it to what's called an ELISA, enzyme-linked immunosorbent assay. And basically, it's the same thing as an IFA, but they automated it so it can be read by a machine, give it a little bit more specificity, or I should say consistency, but it's really not much better of a test. What they do, they take the organism, they sonicate it, and they put it in these little teeny wells. And the machines contain many wells, look like ice cube trays, but dozens and dozens of them. And the machine reads it. So again, it's more consistent, but not a major advance in accuracy. And just like the IFA, it's only one species. Modern testing can include what are called microarrays. There's one commercial lab that uses it, but basically it's an ELISA and uses multiple wells for individual specimens, but still it's single species. Usually it's really not much more accurate based on published studies than the old-fashioned ELISA. In fact, in terms of accuracy, this sensitivity, published studies, 50% average. Specificity is not that great, 70%, 80%. So in other words, 20 to 30% of the time you're getting a false positive, 50% of the time you're not finding when it's actually there. Here's a study that was published in 2007 where 9 different labs were queried and the results were tabulated. And to get a high specificity, 96% or better, this trade-off meant the sensitivity was really pretty poor, 50, 55%, averaged out to 49 from top to bottom. The one I highlighted in yellow, to increase the sensitivity, they had to sacrifice specificity. And the perfect example of that trade-off I said before. So the overall sensitivity averaged out to 49%. A more recent study done by John Orcott and his colleagues, they took patients with DNA PCR positive for Lyme. I mean, they really had Lyme because PCR is, very insensitive but highly specific. These people tested with the standard CDC 2-tier testing, and they went so far as doing acute and convalescent titers. This is said to be the most accurate way of confirming Lyme. Take the first test in the beginning of the infection, wait 4 to 6 weeks and do the test again, and you look for an increase in the ELISA and more bands on the Western blot. Anyway, using this supposedly highly accurate thing, they found that they were wrong almost half the time. They're positive for Lyme only 53.5% of the time and negative all the rest. So again, basically it's a coin toss. How about a Western blot? Well, that's supposedly better, but still it's standard old technology, not recombinant. And the reason why this is different is instead of just like an ELISA saying yes, no, or borderline, here they separate out the individual protein antigens to see which, if any of them, the patient has antibodies to. Again, it's a single lab strain, unfortunately, and by lysing the whole organism, they have many non-specific antigens. You can get some false positive readings. It's also technically difficult to do, difficult to be consistent, and get some false negatives. Here, the sensitivity, again, is 50, maybe in the better lab, 70%, but for that one strain and one species, and specificity, again, varies 80, 90%. Here's actually a picture of a Western blot. And how they make it is they take the Lyme Borrelia that's in a culture, they lyse it, release all the antigens. They separate them out using electrophoresis, electrical charge, and they then transfer this to a membrane strip. And you see in this picture, these little black marks, these are called the bands on the Western blot. So if the patient has antibodies, when they put their serum onto these paper strips, they make these black dots, they bind where the antigens ended up. Okay, so when you do electrophoresis, they're using electrical charge to separate out these different proteins based on their molecular weight. It's not the very exact, it's subject to a lot of technical issues in terms of the temperature of the bath, the salt content, how strong the charge is, blah, blah, blah. And so basically, it's not that precise. And how you interpret this is based on, is a band present or not? If it is, where is it located on the scheme to tell you how the molecular weight? How dark is it? Is it dark and positive? Is it pale and maybe not there at all? So if you look at this far lane on the side here, lane 8, that's a positive control. Now the lab tech gets these, this is lanes 1 through 7, are patient specimens. How do you line this up? If you look up here, like band 60 or 66, where exactly is that in this control strip? How about down low down, 17, maybe below it, 16, I don't know. Very hard to line up, so it's very imprecise. The culture that made this, if it's very strong culture, you get very dark bands. If it's a weak culture, you get weak bands. And how dark does it have to be called positive? What about these here in lane 2? I don't know, some of them might be positive, some not. If you did this test the second time, you might get a different look. So overall, it's, better than an ELISA because you can look at individual bands and make your own interpretation, but still, it's only one species. It's not very precise. Despite all that, the CDC then came up with a scheme to try and standardize the Western blot. And again, this is for surveillance, not for diagnosis. But anyway, that's been used for diagnosis according to them. Now, a lot of problems with this. They required a specific number of bands to be included on the Western blot. They named 10 what they call specific bands, which are not. And to be positive, you need to have 5 of these 10. Okay, and they named these bands. The numbers are up there. Now, the problem is not every patient sample will have 5 bands. So if you have 4 bands, does that mean you don't have Lyme disease? Does it mean you have an 80% chance of Lyme disease? I mean, it's very imprecise and kind of arbitrary. And that's for the IgG. The IgM Western blot have 2 of those 3 bands. But for the IgG, if you look at these numbers, 7 of those 10 are not specific to Lyme. And I put them down there, 18, 28, 30. They're not specific to Lyme. So if you had 5 non-specific bands, you could have a virus or autoimmune disease and be called positive by CDC. So you see, it's really very imprecise. Get false positives. The other problem is bands 31 and 34 are really highly specific to Lyme Borrelia, and they were not included because of conflicts with the vaccine at the time. So besides all that, it's also that lab strain. It's not human strains. It's not the reality of people. So this means the Western blot's not going to be very accurate by the CDC standards. Compare that to an ImmunoBlot. The reason why it's so much better, number one, it's designed to collect or pick up multiple species, okay, the whole list of Borrelia and even more. It's so sensitive that even in early Lyme, the 2 series that were done, one over 83% of early Lyme cases were detected. Another one, CDC's own samples, they detected 93% of early disease. Never before have we ever had a test you can test an early Lyme patient. Some comes in after camping a week or so later and they start to get flu-like symptoms and maybe they didn't have a rash, but something's not right. You can finally now test them a few weeks into it and get a positive 80 to 90% of the time. In the same series, both of those series, in disseminated Lyme, like what they used to call stage 2, they still do in CDC terms, and chronic Lyme, sensitivity was found to be greater than 98%, sometimes even 100%. So the ImmunoBlots basically are accurate enough to be used in all stages of Lyme, early, intermediate, and late chronic Lyme. And this is why they're so accurate. Again, they're using recombinant antigens, these things that are genetically made by reverse engineering from the Borrelia. And instead of using electrophoresis as vague electric charge in a salt bath, they use a kind of a printer to print the bands on the strip. So they know exactly what number corresponds to what antigen. And then printing this, they know how much they printed there. You don't have to depend on a culture. A good, strong culture or a weak culture doesn't have any effect on this because it's not from a culture. So this high precision, and you can see the picture here, this is their control strip. It's available for Lyme, for relapsing fever, Babesia, and Bartonella. Can detect all the major species of all of these different organisms. And because of this technology, it's really the most sensitive serologic test for any of these. And I just put these 2 together. The picture I showed you before, the Western blot, this is the control strip. Compare that to the ImmunoBlot. I mean, you can't compare. It's like night and day, And this is one strain. It's kind of this imprecise, dirty mess. And ImmunoBlot's clean, all these different species and even others. So no comparison. But very important, semantics. I was looking at the LabCorp website, and they said they have an ImmunoBlot for Lyme. So I looked into it. This is the thing. The term ImmunoBlot is kind of an umbrella broad term for tests that separately reports different antigens in a sample, okay? Or reports the antibodies to those antigens. So technically, a Western blot is a form of an ImmunoBlot. And I think these labs are using the term ImmunoBlot, which they only started using last year, to kind of ride on the coattails of IGeneX. So just because it says ImmunoBlot doesn't mean it's the same recombinant multi-species assay that IGeneX offers. So be very careful of this semantics loophole and don't get fooled. All right, so what about the 2-tier testing thing? This is a scheme developed for making a test more accurate, okay? So the idea is you have 2 tests. You have a screening test that's very sensitive at the cost of some false positives. So if it is positive, all right, we'll do a second test that's just as sensitive, but maybe a little bit more specific so you can get rid of the false positives or at least make it better. However, if the first test is negative, then the whole test is done. You don't do the second test, you just call it negative, and that's the end of it. Statistically, for this to work, the first tier must be 99% sensitive, and the second tier should be as sensitive, but also 95% specific. The first question to me is, if the second test is just as sensitive, but more specific, why do the first test in the first place? But that's the way it is. All right, so what about applying this to Lyme disease? So according to the CDC, the two-tier test must begin with an ELISA as tier 1, then use a Western blot as tier 2. They have what's called a modified two-tier test, where you use the ELISA or an IFA as tier 1 and a different ELISA as tier 2. Now you know the ELISA has only a 50% sensitivity in Lyme disease, so right away you're missing half the patients. You would never do that for any other illness. Would you diagnose cancer with a 50-50 test? No. So let's say you get a positive ELISA. Then you do the second test, the Western blot. Because of that restrictive CDC criteria, you're going to also get some false negatives, so you're going to get even less sensitivity out of it. If you instead using an ELISA for tier 2, it's no better than the first tier, so why even do it? It doesn't make any sense. And it's based on that one single lab strain, one species. Now, ImmunoBlot, let me show you the validation here. When you do a combination of IgM plus IgG, you pick up 93% of early cases, 100% of established cases, what CDC calls early convalescent neurologic and Lyme arthritis. And no other test has ever been shown to be that sensitive in early Lyme disease. Now, despite this high sensitivity, from this report, the specificity is between 97 to 99%. These included two different unknowns from CDC, proficiency testing samples, patients' autoimmune diseases, viral infections, even syphilis. And again, 97 to 99% specificity. So here we have a sensitive and specific test. Now, when this went through FDA validation, they tested 304 samples in two different studies, blindly tested. They came from CDC, from IGeneX, from the Bay Area. Again, thank you for that. Tested from endemic areas and non-endemic areas. And the specificity, again, was greater than 99% in all of this. How about reproducibility? This is a blind study to see if you sent this out to different hospital labs or different commercial labs, how accurate would it be if you did the test again and again? So what they did was they ran the test across 3 separate sites, each with 2 operators over 5 days and 2 runs a day, used a panel of samples that were negative, a high negative, a low positive, moderate, and high positives. And guess what? For the IgM, there's 100% agreement every time they did the test across all runs, all days, and all sites. With the IgM, there's no interference from endogenous compounds in the blood. They mixed in low and high doses of albumin, bilirubin, cholesterol, hemoglobin, triglycerides. No effect, 0 effect is seen on the results by doing all this. Then finally they did, all right, let's do a fresh sample versus frozen samples kept for 2 or 3 weeks. 0, no effect at all, 0 effect whether it's fresh or frozen. The IgM ImmunoBlot is interpreted by numbers of bands. So the ImmunoBlots that are FDA cleared contain, it's actually a 2-tier test. It contains a Lyme screen assay, the LSA, as the first tier, and that's right on the same strip. And then the bands. This has 26 bands plus 2 separate controls. And they include, you have to have at least bands in 2 groups, the group 23, 31, 34, 39, and 41. Now, the IgM for the CDC's Western blot is 23, 39, 41. They don't include 31 and 34. Okay, as I mentioned before, 31 and 34 are the most specific bands for Lyme, along with band 39, and that's why this is so much better than the standard Western blot. Now, not only do they have these other bands, they have many of them. They have 9 different 23s, 9 different 31s, 2 39s, 2 41s. Why do they do that? Well, again, it's multi-species. Band 23 might be different for, say, Afzelii than it is for Greenii. So to get all these different species, they didn't use one band, they used multiple bands for each molecular weight. So that shows you why recombinant technology is great. They can just say, I want 26 bands, and not just 1 or 2 or 5. They can do this, and that's why it's so much more sensitive and much more specific. A lot of us have seen clinically people with late Lyme who have persistently positive IgM and maybe a negative IgG. Now, the dogma is that in most infections, the IgM will disappear after 4 to 6 weeks, be replaced by IgG, but we don't see that in a lot of Lyme patients. With standard testing, the IFA, ELISA, even the Western blot, a persistent IgM could be a false positive because it does cross-react with other organisms, especially the DNA viruses, like Epstein-Barr. And this is why the CDC, the IDSA, and NIH tell us ignore late positive IgM in Lyme and disseminate in chronic Lyme because it's probably a false positive. Because first of all, they don't believe IgM persists, they don't read the literature, and that's the dogma. But because the IGeneX recombinant technology allows a high degree of specificity, you do not ignore late IgM on an ImmunoBlot. 99% or higher than 99% specificity in the FDA's own testing. So you see, here the dogma has to change. You don't ignore positive IgM in a late stage patient. How about the IgG ImmunoBlot? They did validation for cross-reactivity, and they looked at all different samples from all different diseases, 376 samples, blind testing, the two-tier method using the LSA and the ImmunoBlot. They found 100% agreement. In other words, they did not find any cross-reactivity with all these other different organisms and diseases. 100% agreement. How about reproducibility? Again, the same thing, blind study, 3 separate sites, 2 operators, 5 days, 2 runs a day, panel of samples, low, medium, and high sensitivity or positivity or negativity. 100% agreement on all test results, on all different runs, all different days, and at all different sites. So again, it's a really reproducible, accurate test. And finally, that's just like with IgM, the IgG ImmunoBlot was tested with different blood substances to see if there's interference. Albumin, bilirubin, cholesterol, hemoglobin, triglycerides tested at low and high concentrations had 0 effect on the accuracy of the test. So there's no interference. The IgG ImmunoBlot is interpreted by finding at least 2 or more bands on the groups. Positive LSA as tier 1, and then 2 or more bands on the ImmunoBlot. And again, it's 23, 31, 34, 39, 41, and 93. To make it be multi-species, just like with IgM, there are 9 different 23s, 9 different 31s, and so forth. So you're getting a lot of different species, you're getting a lot of accuracy. Now, also, remember before I said that P41 on the Western blot is not very specific? Well, that's true because it does cross-react to some antigens, even in nerve tissue in humans. But here, the 41 was derived from the genetic sequence of a Lyme Borrelia germ. So it's different, even though it's the same molecular weight as what's found in humans, it's a different antigen. So here, the 41 is specific, okay? So that's why specificity, 99% or more. Okay, a lot of mumbo jumbo. Does it really matter? I've shown these slides before, but I have to tell you again. These are case series taken from practices of Lyme doctors and practitioners. In this first series are 36 patients, all from North America, who had a negative Lyme Western blot, but they were positive on the ImmunoBlot. What did they find? They find Mayonii, Spilmanii, Californiensis, all these other different species, including Guinea and Afzelii, which are European species, supposedly, and supposedly not in America. Well, guess what? People travel. They can get Lyme disease when they travel. They bring their pets, birds, which are known to transmit ticks and their infections across countries and even continents. You know, they carry this around. And there are published studies showing that, at least in coastal areas, you can find European species of Lyme Borrelia in North America. So you have all these 36. One of them actually was Borrelia burgdorferi strain B31. The Western blot missed it. But they found all these other species that were missed on a negative Lyme Western blot. So of these 36 patients, they all have gotten a false negative result. Interestingly, 5 cases could not be speciated. Those of you who've done ImmunoBlots can see sometimes it says report Borrelia species were found. They don't name which one. Well, what does that mean? Well, it doesn't mean it's a negative test. It's still a positive test. It just can say which one it is. And I asked Dr. Shah why that happens. And she found that in most cases, it's because there's more than one species present. They co-infect with more than one species of Lyme Borrelia. And what happens is you get so many bands that you really have a hard time separating them out. So they just say, well, it's positive. I can't tell you what species, but, you know, don't ignore it. There's another series, 29 patients, North America, negative Lyme Western blots. They found all these other different species, 2 B31s, which mean, again, the Western blot missed 2. Should have picked them up, but did not because it's not such a great test. Here, 11 were unspeciated, and 4 were European Borrelia that were unspeciated. So here we've got co-infections. The third series, I can end with this, 11 cases, negative Western blots, positive ImmunoBlots. Here they found co-infection Spilmanii plus mayonii would not have been picked up at all. One had B31 plus californiensis. So another example of this. Now, because these tests are so good and because the FDA cleared, this has forced the CDC to change its website. When it comes to Lyme disease, it no longer says they have to have 5 out of 10 bands, you have to on IgG or 2 out of the 3 on the IgM. It doesn't exclude 31, 34, nothing. It doesn't say anything about that anymore. All they say is that it has to be a 2-tier test approved by FDA. So the success of IGeneX in doing this has actually won the fight that we've been fighting the CDC literally for decades to update their standards. And finally, they have, thanks to the ImmunoBlot. Now, what does this clearance mean? Commercial labs and hospital labs that do Lyme testing, they don't have a Lyme lab in the corner with a biosafety level 3 hood and grow spirochetes and make all these things. They don't do that. They buy test kits. So how do you get a test kit? A test kit is a thing that the FDA licenses. FDA licenses tests, just like they license blood glucose meters, they license medications, they license things that other people make to use. So labs like that, hospital labs, they buy a test kit and they stick it in their machine, they run it. But what FDA clearance means is that now these labs and these hospitals can buy the ImmunoBlot kits and use that in their hospitals. So if they do that, which I hope they start to do, especially if we start putting pressure on them, they'll finally be able to offer a test that we can trust. So you have to do that. Let the hospitals that you work with and labs you work with know that they can get the Lyme ImmunoBlots that IGeneX created, sold as iDart ImmunoBlots, and they can get them and replace the crappy tests they have now. Now, does that mean with FDA clearance, they're going to be covered by insurance? Well, it's not really clear. There's no rule that just because it's FDA cleared that insurance has to cover these things. But if big labs start adopting it, especially hospital labs where everything's under contract with insurance companies, they're going to be pressured to actually start to cover them. And just so you know, the ImmunoBlots already are covered by Medicare, but we're talking about commercial labs. And I think that the tide is going to turn once these are available because I spoke to at least one hospital system and they're going to be using these and they're already pressuring their insurance company to start covering it. So the story is not yet over. I also want to spend a little bit of time on relapsing fever, tick-borne relapsing fever, because not only is it very common, but it's the same story like Lyme disease. Commercial labs offer tests for Borrelia hermsii. Some of them offer for Borrelia miyamotoi, but look at all the other ones. IGeneX is finding Toccica, Toricate, Coriace, Parkeri, all these other ones in their patients, and they would have been missed, okay? IGeneX testing includes all of these. Commercial labs do not. Now, why is that important? Well, there's a lot of misinformation about relapsing fever, especially the tick-borne relapsing fever that's found in the United States. First of all, it's thought to be rare and just something you find in the Western mountains. Well, that's not true. As I showed you in the map before, I'll go back to that. It's found all across the country, even above the Arctic Circle. How do you like that? Let me go back one more time. This map was generated by IGeneX by county, which counties had positive results in their testing. So this is not some survey of ticks or animals or whatever. This is what they actually got positives on when tested at IGeneX. So it's found all over the United States and around the world. Second thing, it's said to have a very distinctive clinical presentation that could not be missed. That's also not true, and I'll show you data on this, can present exactly like Lyme disease and fool you. Also said to be transmitted only by soft ticks. Well, that's also not true. Many species of hard ticks carry and can transmit relapsing fever. And truthfully, in the southern states of the United States, soft ticks are present even in urban areas. So all this misinformation has caused Lyme doctors to miss cases, and IGeneX is here to help. All right, so what about tick-borne relapsing fever mimicking Lyme disease? Well, if you look in a textbook, classic relapsing fever is a very distinctive acute illness that's very hard to miss. You get a high fever, really terrible chills, severe illness, can last a day or two, even 3, ends in a crisis, severe sweats and weakness. And then the patient feels much better for several days, but 5 to 7 days later, it relapses. The same thing happens again and again. That's why it's called relapsing fever. However, many patients with relapsing fever can present exactly like Lyme disease, and I'll show you why. IGeneX did a case series, 543 United States patients diagnosed by their Lyme practitioners of having Lyme disease, not this classic relapsing thing, but Lyme disease. When they were tested by the ImmunoBlots, 32% had Lyme antibodies. 22% did not have Lyme, but had relapsing fever, and 7% had both. So that's a huge number would have been missed if you only tested for Lyme disease. Here's another report, publication. These are 90 patients who met the clinical published case definition of chronic Lyme disease, were not suspected of relapsing fever. Of those 90, 46 were positive for Lyme and a lot of different species, including Afzelia, Greenii, Spilmania, and so forth. 56, more than half, had relapsing fever and not Lyme disease, and not just Hermsii and Miyamotoi, but some of these other ones. 8 of them actually had both, and this is published. So there are pitfalls to testing for relapsing fever because the standard serologies are not very good. You go to the big commercial labs, you can get an IFA, which is that old technology for Hermsii. Some labs offer testing for Borrelia miyamotoi, and it's an ELISA, but this ELISA is not based on a sonicated Borrelia. They use one protein antigen. It's called GLP. It's like trying to do a Western blot with one band. I mean, it's not very accurate because of that. The other problem is that all those other species are not represented by this. You can have one of those other ones. Let me go back a slide. Hermsii and Miyamotoi, but there's also Tricarte, Trisica, on and on it goes. The other thing, there's a flip side to this. Tick-borne relapsing fever species can express some antigens found in Lyme, and you could think someone has Lyme disease if you use those old tests. For example, you can find band 41 on a Western blot. You can find occasionally band 23. So you can get some false positives on some ELISAs and IFAs because of this. If you get a borderline ELISA or a Western blot, a band of 23, a single band, or a single band of 41, might be a hint that it's not Lyme, but it's actually relapsing fever. So basically what you need to do is test for relapsing fever using an ImmunoBlot. And the data shows that the IGeneX ImmunoBlots don't cross-react. In other words, a relapsing fever ImmunoBlot does not cross-react with Lyme and vice versa. So you really need to order both a Lyme and relapsing fever ImmunoBlot when you're testing your patients the first time. And, the possibility is that the Lyme seronegative patients really don't have Lyme anyway. They have relapsing fever. None of the Lyme test kits that are commercial, the IFAs, ELISAs, Western blots, PCRs, even T-cell tests, none of them for Lyme have been validated for any relapsing fever species. So if you're getting a standard commercial test from one of the big labs or a hospital lab and it's negative for Lyme, but your patient has Lyme symptoms, start to think that maybe it's not Lyme, maybe it's relapsing fever. Relapsing fever validation in disseminated confirmed infections, 100% sensitivity. These are patients with PCR positive, so you know that they had it. Even in early relapsing fever, and early relapsing fever is not like early Lyme. Early relapsing fever is like the first few days when the big fevers start. They still picked up 2/3 of the cases. In terms of specificity, was 98%, including IgM once again. It was better than 98%. So here we have, again, high sensitivity, high specificity, and it's useful in early infections. I'll give you some case series. I don't want to beat a dead horse, but just to show you this is reality, not just some lab fluke. Here, first 12 relapsing fever patient samples were tested by IGeneX, by the ImmunoBlot. They found Tricarte, Miyamotoi, Hermsii. One had co-infection of Hermsii and Miyamotoi. 48 cases in the second series, they found not just Hermsii, Miyamotoi, but Tricarte, Trisica-like. 26 unspeciated, which means they probably had co-infection. Third series, this was not America alone. These were 35 patients, including Europe and Australia. They found a lot of different ones, including 16 that were unspeciated. So this is real, guys. You really have to think relapsing fever when you test your Lyme patients. I want to mention babesiosis because you can get an ImmunoBlot for that. Lab data shows it's the most common co-infection in Lyme patients. Being that it's a parasite and not a bacterial virus, you have to think completely differently in terms of testing. But it is transmitted by the same tick that transmits Lyme. So again, you have to think about it, especially because a lot of symptom overlap. But here's the thing. When Lyme patients are concurrently infected with Babesia, they're sicker. The symptoms are more severe. The immune system is more compromised. Lyme treatments need to be more aggressive and given for a longer duration. This is published data. So when you have a sick Lyme patient who's really sick and not responding well to treatment, you have to consider babesiosis. Even sometimes they don't present with the classic, sweats, day sweats, night sweats, and so forth. Because, the immune system may be so low, they're just not generating the response you expect. So when you have these really, really sick Lyme patients, test them for Babesia. You'd be surprised how many you find. Same story, just like Lyme, just like relapsing fever. The common species B. microti is what the labs will test for. Some labs you can get done canine, but that's it. You can't even test for these other ones. Can you get a test for bigeminal diversions, all these other ones that are being found? You can't. IGeneX testing does include all of these. And here about accuracy, no cross-reactivity. Here's the first series. I tested Babesia ImmunoBlots in patients with Lyme B31, as well as a human strain 297, relapsing fever, Bartonella, E. coli. The second series against patients Anaplasma, Ehrlichia, Rickettsia, Lyme, and normal controls. Specificity is 100% in all these things. So you get a positive Babesia ImmunoBlot, they have Babesia. Sensitivity is really very high. Now, here's a very interesting thing I found. Here, whoop, sorry, I moved over. Where am I? All right. 63 samples that were positive, 25 had microti, and 20 had Duncani. What were the other 18? Okay, these are other species that were making the patient sick that showed up on the ImmunoBlot that you could not otherwise find in any other lab. So again, it's important to do the ImmunoBlot when you suspect Babesia. Same thing with Bartonella. You can see on the map, again, these are positives that IGeneX found in America, including Hawaii, Northern Alaska, North, South, all over the place. But it makes sense because, why is it so common? You can get the infection from a lot of different vectors, fleas, mosquitoes, biting flies, mites, red ants. Proven now you can get that ticks can harbor and transmit Bartonella. Of course, animal bites and scratches, needle sticks, veterinarians have had that happen, maternal fetal. Over 45 species of these are out there. At least a dozen, if not more, have been documented in human infections. So it's really something important to consider in your patients. The Bartonella ImmunoBlot, just like the other ImmunoBlots, very broad species coverage. They can identify Elizabetha, Hensley, Quintana, and Vinsonii, and they'll name those, but it does also pick up the other ones. This does not cross-react to other tick-borne diseases. Sensitivity and specificity, when you look at the other tick-borne diseases, was really very, very high. The specificity was 100%. And sensitivity was, when you combine IgM and IgG, it picked up all of them in this case series of other tick-borne diseases. So it's really a good test. And we know how Bartonella is such a stealth organism, so hard to pick up. So this is actually pretty good news. So I'm pretty much at the end of this. I have some closing thoughts. We always talk about tick-borne disease as being a clinical diagnosis. But in today's climate, more than ever, you really need a good lab to back up your clinical diagnosis. And here we have that opportunity. The FDA clearance, as I mentioned, has provided a window into the extent of validation that the ImmunoBlots went through. I mean, all this different testing at different labs, different people at different times, frozen, not frozen, different interfering substances. All this is what they went through. But the thing is that they have done this anyway when they developed these, even before FDA, and they do that for all the ImmunoBlots. And what I find really incredible and also reassuring is that to go through FDA, they didn't have to make the ImmunoBlots different or better or change them or whatever. This is what they have. This is their ImmunoBlot, the one that we've all been using all this time. So it's really reassuring to see that it can pass muster with the most stringent of testing. So I really think it's why you need to do IGeneX when you do your testing. It's proven to be the best. With FDA clearance, now you can pressure your local labs to do the iDarts and get rid of their old-fashioned low-tech Lyme tests. And that's it. We're done with the presentation, but we will be available now for questions and answers. Thank you very much.